Hello, and welcome to this Ethical Dilemma in Science, where we're going to take a look at the science and ethics associated with cord blood banking. And we ask the question, is cord blood more than just blood stem cells? And so for this scenario, what I want you to consider is that a close friend or a family member who is about to give birth is asking your advice about cord blood banking. This is a process that is a little over 30 years old now. It's basically taking discarded perinatal tissue associated with the birth, and storing it in a way that it can be used in the future. And there are a number of both private and public banks associated with collecting cord blood. Now, if we take a look at this, generally what we're focusing in on is kind of remnants of the placenta and the umbilical cord. And so from a biological standpoint, the placenta is a combination of maternal tissues that are formed by the mother, as well as fetal tissues that are formed by the fetus early on in development. And the combination of the placenta, both the maternal tissues and the fetal tissues, kind of come together and allow for kind of support of the embryo and the fetus as it's developing up to the end of gestation. But what scientists have learned is that within these placental tissues and within the blood that's found within the umbilical cord at the time of delivery, there are going to be a variety of cells that can be isolated and used in future settings. Traditionally, and again, tradition 30 years old, but basically it started out collecting umbilical blood stem cells. Cells that I'll define over the next couple slides have the capability of developing into adult cells, blood cells that may be needed later on by the individual. More recent evidence has determined that there are a variety of other types of either progenitor cells or stem cells that are found within these tissues that are normally discarded. And so the question then becomes, is it going to be appropriate to collect this tissue? And when they do that, they collect a medical history of both the mother and the infant. They'll often characterize the cells so they know exactly what it is that they've collected. But ultimately, they're going to store them for future use. So generally, what's going to happen is they're either going to be involved with a public bank or a family bank or a private bank. Now, in the private banks, which this slide is describing as the family bank, basically the family owns the cord blood. They're going to pay a fee up front to kind of collect the tissues, isolate the tissues, and store the tissues in an appropriate way that they can be used in the future. But in general, what we're going to see is that the blood stored within a private bank or a family bank is under the control of the family. And so it could be used for related transplants. So a family member that needs cells or tissues that could potentially be derived from these blood cells, these blood stem cells, or potentially it could be used for stem cell therapy for other related uses, mainly for the individual that has donated the tissues. Now, in contrast, these public banks are collecting the blood, in some cases kind of minor fees for the individual, but in general, many of the fees are going to be waived by the public banks so that they are able to collect these tissues, these cells and tissues that are available for matching kind of in the public, matching kind of across the community, with the goal being that they are going to provide a source of cells for individuals that need it later on. Now, as this technology is emerging, regulations are kind of racing to catch up with it. Over the last 10 years, mainly since about 2009, the Food and Drug Administration, the FDA, has defined these biologics, defined these cells as being both a drug and a biological product. And so because of that, it's requiring that these public banks, which are collecting these tissues and potentially making it available as a medical treatment for a wide range of individuals, not just the individuals that are directly related to the original donor, they need to be able to kind of go through the process that the FDA has for ensuring that the tissues are collected and maintained in a safe way, that they're not getting contaminated, they're not being damaged in any way, that they're going to be viable and appropriate for use later on. Now, this is probably a good idea. You want to make sure that the cells are going to be viable and useful at some point in the future. But seeking that license becomes very difficult and very expensive. And because of that, many of the public banks have difficulties obtaining this licensure, obtaining this certification. And because of that, limits their ability to use the tissues that they've got stored within them. The other thing is that licensure, these FDA licensure to be able to use these tissues, isn't retroactive. And so previous samples that have been collected are not normally covered under the ability to use them as future treatments. And so that, again, provides some level of difficulty in this area of potential cell replacement therapy. Now, in order to understand what these stem cells are doing, we need to be able to understand what cells in your body do normally. Now, in the process of development, you're going to start out with the equivalent of a fertilized egg. And that fertilized egg, composed by the egg cell from the mother and the sperm cell from the father, coming together and forming that new individual, a new, what would be a zygote is the technical term, but what will eventually develop into the fetus. Now, as those cells develop and as the fetus develops, it becomes more and more specialized. So the cells start out being able to become virtually anything within the body. And so we've got the term of totipotent stem cells is being something where we have the opportunity to produce both the fetal tissues, what would develop into the infant that's born, as well as portions of the placenta. But over time, these cells are going to become specialized. They're going to start to become more focused and more, the term is differentiated, for doing certain things. So after a period of time, those totipotent stem cells lose the ability to produce portions of the placenta, and they become what are referred to as pluripotent stem cells. Still lots of potential because these cells are the rough equivalent of embryonic stem cells, and they can develop into any of the fetal tissues. And so in theory, you could take one of these embryonic stem cells and grow a new individual. It's very difficult to do in practice, and it's not, it's science fiction now at this point, but potentially at some point in the future. This level of specialization and differentiation occurs, and so that when we're looking at cord blood and the stem cells that are associated with the cord blood, what we're looking at are going to be referred to as multipotent stem cells. These are going to be stem cells that are referred to as hematopoietic stem cells, which is a fancy way of saying these are stem cells that can develop into and produce new blood cells. In addition to these terms, more recently, there have been studies that have looked at induced pluripotent stem cells. These are basically cells that are collected from the adult that have been treated in a laboratory setting, in a clinical setting in some way, so that they are made more embryonic-like. They're made less specialized. They're made into, as it said, induced into these pluripotent stem cells that could then potentially be pushed into the development of other types of cells that are needed by the body. Now, the problem with that, with these induced pluripotent stem cells, is we are starting with an adult cell and kind of pushing the clock back, pushing it into this undifferentiated state in the hopes of kind of restarting or mimicking the normal developmental process. This is an area that requires a lot of research going forward. Now, if we take a look at these, again, these multipotent stem cells, these hematopoietic stem cells that are found within the cord blood, what we're going to see is that basically they have the potential to continue to divide in the laboratory setting or the clinical setting. And some of those cells are going to start to become specialized. Some of them are going to start to, again, the term is differentiate. They're going to start to differentiate or become specialized into distinct cell types that are used within the body. And so some of these cells are going to develop into the red blood cells that are involved with carrying oxygen throughout the body. Other cells are going to develop into lymphocytes, which are involved with your immune system. And still other cells are going to develop into a whole range of other white blood cells, which are involved in protecting your body in a variety of ways. And it's going to be important that you have these functioning blood cells throughout your lifetime. If you don't have functioning red blood cells, you're going to have a lot of difficulty getting oxygen to the cells that need it throughout your body, and there are disease processes associated with that. Diseases associated with the lymphocytes are associated with kind of growth, abnormal growth or cancers of these white blood cells are things like leukemia. And the normal treatment for leukemia is this idea that you find a donor with the appropriate match for the characteristics of the recipient. And so you collect the stem cells from the donor, possibly process it in some way, and then ultimately they're going to be transplanted or injected into the patient. But in the interim, what happens is that patient is going to have their cancerous cells destroyed. The disease cells, in essence, destroyed, so that when they get those stem cells in, the stem cells don't have to compete with the cancerous cells, and they're able to basically become established within the individual and reform kind of a functioning blood system within the body. Now, the ability to use this cord blood in these cord blood stem cells means that we can bypass the need of identifying a donor and potentially be able to transplant in these tissues or these blood-forming cells, these blood-forming stem cells, from an individual that has banked their blood, or somebody as a close relative of an individual that has banked their blood. Now, in addition to the approved uses, the FDA-approved uses for these cord blood stem cells are mainly for the treatment of blood-borne diseases. But there are over 1,100 clinical studies out there now that have either been started, are in the process, or have been completed that have looked at cord blood transfusion for a variety of conditions. Now, a majority of these are associated with blood diseases because it makes sense that these blood stem cells would be used for the treatment of adult blood cell diseases, or even child blood diseases. But it's been determined that there are a whole variety of other sources or other uses of these cells that are possible. And so these non-blood-based replacement therapies have focused in on areas like the nervous system. And so there have been studies, and there are now clinical studies that are ongoing, looking at the transplantation of these cells in for the treatment of ischemic stroke or traumatic brain injury. There are other studies looking at metabolic disorders like diabetes, looking at endocrine diseases that can potentially be treated with these cells. And so we can see that there's a lot of potential associated with these perinatal tissues, these tissues that are present at birth, that could potentially be used to repair or as a treatment for a lot of biological disease conditions later on. Still, this gives rise to a number of ethical dilemmas. So the first one is, how do we get more of these stem cells? Most of the adult therapies that use cord blood transplants require 2 units. And so they have to take the equivalent of 2 donations, a donation from one individual and a donation from another individual. And so the question becomes, should scientists then explore ways in which, like they did that induced pluripotent stem cell that I mentioned earlier, the adult cells that have been induced to become more stem cell-like, how about if we figure out a way that we can de-differentiate or unspecialize these blood stem cells to move them back a couple steps so that they are able to divide and produce many more cells. And this then could potentially allow us to have a recipient to receive all of the cells from a single donor rather than requiring 2 units or donations from 2 different individuals. If we extend that a little bit further, we know that there is a lot of interest in the use of embryonic stem cells, but the embryonic stem cells have a lot of controversy associated with it because they have traditionally been derived from aborted fetal tissues or from unneeded embryos formed through the process of in vitro fertilization. Now, if the scientists can't get these blood stem cells to divide and potentially become less specialized to de-differentiate and divide to produce more cells in the culture, perhaps we can push them back even further and make them the rough equivalent of these embryonic stem cells. And then from this population of cells, we could potentially generate lots of other types of adult cells that are needed. And so is this something that could be doing to mimic the effects of embryonic stem cells, but deriving them from a non-embryonic source? And then finally, if we have this ability to produce these specialized cells, what else can we do? Right now, we scientists and clinicians and in clinical laboratories are using biological chemicals to treat these cells and push them in one direction or another direction, to push them to become specialized to do certain things and develop into certain types of cells that are needed within the body. Now, many times we understand those chemicals, we understand those signals, and so we've got a good understanding how to do that. But there are other times in which there are interactions that are required between cells in order for the cells to develop properly. The technology is coming out that we can start to develop and grow these stem cells under conditions that allow them to form like the equivalent of like little artificial embryos. And these artificial embryos, called embryoid bodies, can mimic early stages of specialization and differentiation. And so we have the ability then to combine these cells in a controlled setting, form these kind of artificial embryos, and allow them to develop to a certain stage and have that inherent kind of natural process controlling specialization and differentiation. But if we do that, should there be limits on how far development is allowed to proceed? Since these cells, in essence, are acting as clones for the donor. And so these are just some of the kind of controversies and ethical questions that need to be considered as we're looking at greater application of cord blood stem cells. So thank you for listening to this ethical dilemma. As always, if you have any questions, please reach out to me. Thank you and have a great day.